Devo stands for the field of evolutionary developmental biology, which applies an understanding of development from a fertilized egg to a mature individual, to evolutionary biology. And it's a very exciting area of biology. Evolutionary developmental biology helps to explain how changes at the molecular or genetic level can lead to reproductive isolation or can result in the relatively rapid changes that are characteristic of punctuated equilibrium. We will look at some examples of how changes in specific genes like BMP4, gremlin, PAX6, or the Hox genes can result in those rapid significant body changes. Developmental genes control the rate or patterns of development of morphological features like arms, or in this case, digits, your fingers, your toes. During early development, the feet of chickens and ducks are very similar. See the illustration there. The BMP4 gene codes for a protein that initiates apoptosis, or controlled cell death. By activating BMP4 in the developing foot, the cells forming the webbing right here between the developing bony digits undergo apoptosis and die, removing that webbing. In ducks, though, the gremlin gene produces a protein that acts as an inhibitor of the BMP4 gene. It is expressed in ducks to prevent apoptosis of the webbing, but not in chickens. One change, the activation or inhibition of gremlin in the webbing cells, results in two very different foot phenotypes. A change in the foot phenotypes between clawed and webbed for ducks makes a big difference in their ability to be successful in their aquatic environments. In this example, we don't see intermediate forms. It's either a webbed foot or no webbing. Hox genes are another group of developmental genes. These are homeotic genes which code for transcription factors that will turn on other genes. They are important for creating the head-tail axis of the body and controlling differentiation in different regions of the body. So determining what organs form where. The pattern that we see is that there are fewer Hox genes in simpler body types. Mammals like a mouse or human have more Hox genes than the body plan of a fly, which is relatively simple. Evolution of more complex body forms seems to have involved the duplication of ancestral Hox genes that then specialized to turn on genes in different locations of the body to create that main axis and differentiate the body parts. So this illustration shows our ancestral genes shared by Drosophila, the fruit fly, and a mouse and a human. The more complex body plan of the human comes from duplication of this gene and then specialization in the different regions of the body so that the genes are turning on other genes to create the structures associated with the head, the thorax, and the abdomen. Heterochrony involves a change in the rate of development of different body parts. Relatively large changes in the phenotype can be made by small changes in the rate of development. The illustration on the right here shows that with a chimpanzee. If we start with the fetal skull, here's our fetal skull, development through infancy to adulthood involves changing the rate of development. This part of the skull grows relatively slowly. This part of the skull grows quickly, giving us our adult skull. Pedomorphosis is the retention of juvenile traits in adults. We see that pattern in the human skull. If you compare the infant skull of a human with the infant skull of a chimpanzee, both are characterized by a rounded skull and small jaw. In humans, a small change in developmental rate, particularly in the bones of the jaw, results in an adult phenotype that is still characterized by a rounded skull and relatively small jaw when you compare it to the adult skull of a chimpanzee. The eye really is an incredible organ. One of the genes involved in eye development is PAX6, which is a regulatory gene that codes for transcription factors that will then turn on other genes. PAX6 is an ancient gene that seems to have evolved around 500 million years ago and is shared by nearly all animals. The ancestral gene controlled other genes, so it turned on other genes that worked to produce a primitive eye, essentially a pigment cell and a photoreceptor cell that could respond to light. The evolution of more complex forms of eyes resulting from selective pressures to respond to different amounts of light or different wavelengths so we can see color, or visual acuity so we have a sharper vision, resulted from duplications of the PAX6 gene and changes to the PAX6 gene. You can see fruit flies have their version of PAX6 and mammals have their version of PAX6, as well as changes to genes that are also controlled, that also control eye development. However, it's interesting that all of those genes that control eye development are still under the control of the PAX6 gene. Let's summarize with our big ideas. Relatively rapid changes in speciation can occur from changes to developmental genes. Devo stands for Evolutionary Developmental Biology and attempts to understand how genetic changes can drive speciation and morphologic change. That concludes Chapter 25. Our next lecture is our case study. I'll see you there.